Hello all, this video will be explaining the first part of the first topic for biology in Year 9, tables and graphs. First of all, we need to understand what bits of information from an experiment corresponds to what variable, so that we can successfully put it on a table or graph. When you look at an experiment, there are the independent and dependent variables. Independent variable is one you change, and dependent variable is the one you measure. For example, let's say we look at how much a plant grows, depending on how much sunlight it gets. The amount of sunlight it gets is the independent variable, because you are changing it. And the amount the plant grows is the dependent variable, because it is the one we measure. Also, in an experiment, you also want to keep some factors the same, so you won't unfairly change the results. These factors are the control variables, sometimes called the confounding variables. For example, in the plant experiment, the control variables might be the amount of water the plant gets, the type of soil the plant is in, etc. You can use this memory technique. Ice cream, dairy milk, chocolate sauce. Independent variable, change. Dependent variable, measure. Control variables, keep the same. If we wanted to make a table with data from an experiment, we would put the independent variable on the left and the dependent variable on the right, as shown on the diagram. Usually experiments have multiple attempts, so make sure to include them by writing something like Test 1, Test 2, Test 3 below the dependent variable title. Then, in each attempt column, write the data in. If you have multiple attempts, make sure to also have an average, also called the mean. This is shown on the diagram as well. Also, any anomaly should be excluded from the mean. Lastly, remember to include units, but only in the titles. What if you need to make a graph out of the data? First, you need to decide whether the data you have is continuous or discontinuous. Continuous data means data that could be any number, however specific, on a scale. Some examples of this are height, weight, speed. When talking about variation, genetic and environmental factors affect this. It is shown by a line graph. Discontinuous data is data that goes into distinct categories. Examples are blood group, gender. When talking about variation, only genetic factors affect this. It is shown by a bar chart. To make a graph with continuous data, put the variables to their respective axis. Independent variable goes on x-axis, and dependent variable goes on y-axis. Put the data in. Make sure at least 50% of data fills the graph. Give a title, and make sure to put units with a text describing the two variables next to the axis lines. Graphs with discontinuous data needs a title, units, and make sure for the x-axis, it has the different categories, and whenever you put in data, it should go up the y-axis in a bar. Lastly, scatter graphs. A graph with data in it either has a positive, negative, or no correlation. We can determine this by looking at the graph and seeing which way the data is going. We can also describe by how much the data is scattered out by saying it has a weak or strong, positive or negative correlation.